So distinguished guests and esteemed colleagues, ladies and gentlemen, it's really a pleasure to welcome you for the celebrations of the International Day of Women in Radiopharmaceutical Sciences, which is organized today. The seminar is organized today under the auspices of the European PRISMAP project. For your information, this webinar will be recorded and posted on the PRISMAP website. And you can, all through the webinar, also ask your questions in the chat box. My name is Sarah Batu, and I'm the recent appointed Deputy Institute Director of Nuclear Medical Applications at the Belgian Nuclear Research Center, SCKCEN. And together with my partner in crime and co-moderator for this event, Professor Renata Mikołajczak, and our panelists, we have prepared for you a webinar that we hope will be very stimulating your interests. But we propose to start by asking Renata and our panelists to shortly introduce themselves. So I would like to ask you, Renata, to start with, that you can
Thank you very much. Yeah, thank you very much, Sarah. And good afternoon, everyone. My name is Renata Mikołajczak, and I'm working at the Radioisotope Center Polatom at the National Center for Nuclear Research in Poland, with my experience in radiopharmaceutical development, radionuclide production, etc., and also supporting women in pharmaceutical sciences. Thank you.
Thank you, Renata. Professor Ulla Engelmann. Ulla?
Yeah, hello, everybody. I'm Ulla Engelmann, and I'm since the beginning of the year the director for the JRC's Directorate on Nuclear Safety and Nuclear Security. And the directorate is based in Karlsruhe, but also in other European sites, like in Geel, in Petten, and in Ispra. And the link to radiopharmacy is that we have JRC Karlsruhe with a pioneer in the targeted alpha therapy, and we are also looking into strengthening the European supply chain for radioisotopes with our colleagues working on different projects, and some of them are also here in the webinar today.
Thank you, Ulla. Thank you very much. Dr. Dana Nicolai. Dana, can you hear us? We can not hear you, unfortunately. Maybe you can try to connect again. Let's move to Dr. Christina Soberg-Pedersen.
Yeah, hello. My name is Christina Soberg-Pedersen. And I'm working as a radiochemist at DTU, the Technical University of Denmark. Yeah, and I will give a short presentation later. So yeah.
Thank you, Christina. Dana, Dana Nicolai, can you join us again? Can you hear us? Oh, difficult. So let's move to Mrs. Kat Spoormans.
Hi, I'm Kat Spoormans. I'm working at SCKCEN in collaboration with KU Leuven right now on a dosimetry topic, and I studied also physics before at KU Leuven. I will introduce you my topic later also.
Thank you, Kat. And I think Dana is back with us. Dana, do you want to try if your microphone is working? No. So we try a little bit later, Dana, don't worry. So let's move to the agenda of today for this webinar. So you see that it's quite an interesting agenda. We will give first just a small introduction about what is PRISMAP, what is a PRISMAP project. A little bit of the history and the goals of this special day, of the International Day of Women in Radiopharmaceutical Sciences. Then we will move to some pioneering women in the field of radiopharmaceutical sciences and the groundbreaking research that they have performed. Then we have chosen 3 showcases, 3 ladies who are going to present and introduce their research, not in full depth, but to give us a kind of an idea of the type of research that you can do in the radiopharmaceutical sciences. Then we will have a debate about topics of interest related to diversity, to inclusivity, to bridging the gender gap, and also some specific gender-specific health issues. And we will have at the end a Q&A session, and we will try to tackle your questions and your answer at the end of this webinar.
Sarah, sorry to interrupt you, but the presentation is not moving. We are still in the first slide.
I'm really sorry. You missed this second slide with all the nice pictures of the panelists. So then we move to the third slide, which is the agenda of today. As mentioned, we will speak a little bit and introduce shortly the PRISMAP project, then the history and the goals of today. We'll move then to some examples of pioneering women scientists in the radiopharmaceutical science. And then we will have 3 showcases, 3 ladies, 3 female scientists that will explain their research and their clinical practice. And then at the end of the webinar, we'll have a debate about topics of interest and that are gender-related, diversity, inclusivity, bridging gender gap, and gender-specific health issues. During all the webinar, you can ask your questions in the chat, and we will try to also have enough time at the end of the webinar to tackle the questions that you would have. So a short introduction about the PRISMAP project. So PRISMAP is a European medical radionuclide program. And as we know, nuclear therapy and molecular imaging are widely used in hospitals for new promising medical procedures, and they can drastically improve the outcome of many medical conditions and enables the treating disseminated cancer in particular. However, one of the main limiting factors to the development of radiopharmaceuticals has long been the limited and the difficult access to radionuclides that are not yet commercially available. So PRISMAP can count on a large number of highly reputable partners and institutions that are spread all over Europe. So PRISMAP is there, in fact, to support the ongoing research across Europe and beyond, and to provide the immediate access to various novel radionuclides that you can find in this slide. So quite a numerous number of facilities that you can see on these slides are helping to provide new radioisotopes and also to open their infrastructures for scientific as well as for preclinical research. So you can access to the radionuclides that are produced by these infrastructures by submitting a proposal to one of our calls. And guess what? There is an open call for proposals that just opened last week. And so on top of the list of radionuclides that are already available on the top left, you can see you have also another extra 2 new radionuclides that are now also available. So through this call, you can have access to the production and to the delivery of high purity grade radionuclides for medical research. So to also a selection of biomedical research laboratories to perform your associated research and also your preclinical research in a kind of self-service or in a fully performed as a service manner. The deadline for the submission of the project It is the 29th of September, and if you have any questions, please do not hesitate to contact the PRISMAP help desk if you would like to apply, and we hope that you will be numerous to apply for this call. It looks like I've lost again the slides. Is it possible, or can you see my slides about PRISMAP?
We can see the presentation, but open on the first slide.
Again,
so
yeah.
Try again.
It's very bizarre. Okay, I'll try again. Okay, so now I'm back to slide where I was. So on the introduction on the International Day of Women in Radiopharmaceutical Sciences, about this special day. So it's a day that has been kind of created by the International Atomic Energy Agency to recognize a momentum occasion to the invaluable contributions of women in this field, and which aims to highlight the need also for greater inclusivity and diversity within the radiopharmaceutical community. So radiopharmaceutical sciences, we know, play a pivotal role in advancing nuclear imaging, diagnosis, and therapy. And through the use of radiopharmaceuticals, we can unlock a wealth of information about the human body and enabling us to detect diseases at their earliest stages and to guide targeted treatments. So the impact of radiopharmaceutical sciences on patient care is immeasurable, and it is crucial that we acknowledge the instrumental role that women have played in shaping this field. So I would like to start by asking Renata to give us a kind of a screenshot of some pioneering women who have paved the way in the field of radiopharmaceutical sciences.
Thank you, Sarah. I will give you a few examples of women active in radiopharmaceutical science. When we talk about this topic, first of all, Maria Curie Skłodowska comes to our mind, but there were several other female researchers who contributed a lot to develop our field. And the last but not least is Joanna Fowler, the lady who contributed to the development of FDG and its applications in human. So history is created today, and you can learn more about that in a nice paper written by female authors in nuclear medicine and biology on women in radiopharmaceutical and in radiochemical sciences. And next slide, please. In fact, these three ladies here in the top right corner, they were editors of a full issue of Nuclear Medicine and Biology with several scientific papers by female authors. I wanted to mention that we had similar ideas within PRISMAP, and our mini review on copper-67 was also written by female authors, and I'm very proud to be part of this work. Next slide, please. Today we have several women being very important for the field, and I would like to mention here just a few of them. Jolanta Kunikowska, who was served as the EANM president in 21-22, to mention here that she was the second female president of EANM in its over 30 years history. I would like to mention Dr. Aoud, president of Women in Nuclear, in Nuclear Global. And also Melissa Deneke, who is currently the director of IAEA Division of Physical and Chemical Sciences, the division which is coordinated most of the collaboration works within our field. And there are certain organizations within these institutions. Like at the INM, we have Women's Empowerment group, and Evangelista is leading that group at the INM. And slide, please. And what has been mentioned already by Sarah, that our event today, our webinar, is somehow a consequence of the initiative undertaken by the IAA on networking women in radiopharmaceutical sciences. This is part of the Women in Nuclear Global, and we had the first meeting of this network in 2019. Now the group is actively promoting women in radiopharmaceutical sciences. On the IAA side, this is Dr. Aruna Korde, who is coordinator of Women in Radiopharmaceutical Sciences. And I wanted to mention also that Aruna Korde is on the advisory board of PRISMA that brings us all together today. Thank you very much, Sarah.
Thank you, Renata. Thank you very much. And now we will move to 3 showcases to try to illustrate in a nutshell the importance of female scientists in research and in the clinical practice of radiopharmaceutical sciences. And we will give the floor to Christina Sobok Pedersen to start with, and she's coming from DTU Denmark. Christina?
Yes, yeah, thank you very much, and thank you for the possibility to present at this event. So I will try to give a bit of an idea about what kind of research that I'm involved in, and it will be mainly about production of radionuclides for medical applications. Next slide, please. So my background is in chemistry. I studied at Aalborg University, also in Denmark. Afterwards, I did a PhD in radiochemistry at DTU, working with separation of radiometals and also some synthesis of radiotracers. I actually stayed at DTU after my PhD, first as a postdoc and now working as a radiochemist. And now I'm getting a bit more into the production of radionuclides. So I think the great thing about our field is also that it's very interdisciplinary. So I'm learning a bit of physics now, so that's quite exciting. Yeah, next slide, please. So the Heavy Lab where I'm working is part of DTU Health Technology, and we are both doing like research in radiopharmaceutical sciences, but we also have a GMP facility that produces mostly fluorine-18 and copper-64 radiopharmaceuticals for patients. And we are part of the PRISMA consortium, both as a radionuclide production facility and a biomedical facility. Oh, and on the picture you can see we're actually expanding our facility right now with a second cyclotron, or new cyclotron. So yeah, it's quite exciting to be part of that. Next slide, please. So my presentation today will mostly be about radiometals. And the reason why we're interested in radiometals for medical application is because there are so many different radiometals with a large variety of physical and chemical properties. There's both radiometals that can be used for medical imaging, for PET and SPECT, but also for therapy, like radionuclides that emit alpha, beta minus, or Auger electrons. And yeah, part of our research and development is also to make these radiometals more available which is also part of PRISMA to increase the research in many different radiometals and studying their effect. So in order to use a radiometal for radiopharmaceutical, it needs to be attached to a targeting vector which is biological active, which, for example, can trace or find cancer cells in the body. And the radiometal is attached to this targeting vector with a bifunctional chelator. And I will today mostly focus on the radiometal and a little bit about the labeling as well. Next slide, please. So as an example, I will talk a bit about lanthanum-135. It decays by electron capture and emits some Auger electrons and a few gammas as well. But it has a suitable half-life for radiopharmaceutical, and it can be chelated by DOTA, which is a commonly used chelator for radiopharmaceuticals. And the Auger therapy is very interesting because the Auger electron deposits the energy in a very short tissue range. So if it's possible to get the radionuclide very close to the DNA of, for example, cancer cell, it will be a very accurate therapy and have very low damage to the healthy tissue. Next slide. In our lab, we produce the radionuclides using cyclotrons. Right now, we have one of these PET tray cyclotrons with a medium low energy. And as I mentioned in the beginning, we are getting a second one now. So in the case of lanthanum-135, we irradiate a target containing barium-135 with protons. This creates a compound nucleus which is very short-lived, and a neutron is emitted, forming the lanthanum-135. So this is the PN nuclear reaction, which is the most of the radionuclides that we can produce with our cyclotron are formed with this reaction. Now the challenge is that the target material that we irradiate is often present in like 100 milligrams. And even if we produce high activity of the radionuclide, it's only present in very, very small amounts. So we need an efficient way to separate the radionuclide from this target material in order for it to be useful in a radiopharmaceutical. Next slide. Thanks. So for the lanthanum target, we first prepare the target by pressing a powder into a silver disc using a hydraulic press. The silver disc is then put in a target holder, which is cooled by water on the back, and we have a foil in front of it. It degrades the energy a bit, but also makes sure that the target, because it's a powder target, that it is well protected. And on the last picture, you can see a target after irradiation, and this is actually one that looked quite nice, we think. So yeah, next slide. So after the irradiation, the target is dissolved in hydrochloric acid, and we then have this mixture of the radionuclide and the target material. We can actually reuse the target material by precipitating the barium, and the lanthanum is then purified using a column containing different kinds of resins, which have functional groups that binds the lanthanum efficiently, and the impurities and target material can be washed out, and then we can elute the lanthanum in a high purity. Next slide, please. So at the end, we analyze our product with gamma spectroscopy to see if there's any radionuclidic impurities in the final product, and we use ICP-OES to quantify non-radioactive metal impurities, because they can interfere with the labeling afterwards. So sometimes we also do different labeling studies with different chelators to see how efficient the labeling works. And we can also do stability testing of these complexes, for example, in serum, to evaluate which kind of chelator would be suitable for a radionuclide. And during this process, we were able to get lanthanum in an amount and purity that we think is acceptable for preclinical studies. But yeah, this is just kind of the beginning of a radiopharmaceutical production, but I hope it gave a bit of an idea about what I'm working in. And yeah, thank you.
Thank you. Thank you so much, Christina. It was very, very clear. Thank you. And we might have some questions for you, but at the end of the whole webinar. I hope that in the meantime, Dana has managed to reach us and to connect again to the webinar. Dana, can you give us a sign?
Can you hear me now?
Yes, we can hear you. Wonderful.
That's great, and sorry for that. There were some safety requirements. So I will introduce myself now. I am Dana Nicolae. I am heading the Radiopharmaceutical Research Center, where we are doing radiopharmaceutical drug development from radioisotope production to clinical trials. Next slide, please. Beside of my, happy days when I can be senior scientist, I am most of the time doing management of the Radiopharmaceutical Center, and of course in the spare time I am also doing production. But I think the more important for this webinar are my involvement in European Association of Nuclear Medicine Radiopharmaceutical Sciences committees, and also with EIA, trying to help as much as possible with many type of events in our field, which is really much more complex than working in the lab. Next slide, please. So our main development interests are in development of and translational research of PET and SPECT radiopharmaceuticals. Next. As infrastructures, we do have a variable energy cyclotron, which allow us for production of big 4 PET radioisotopes, but also some radiometals as 64, zirconium 89, and gallium 68. These are, let's say, our favorites, and we do some customized extension of the beamline, where we can host a fully automated solid target for metal production. Next, we also have a full GMP radiosynthesis and clean rooms for EU GMP license production. Next, please. And of course, we also are trying to do our best in translational part, starting from radioisotope production to radio labeling and purification, to do the dispensing, and also marketing authorization or clinical trials, depend on the product. Next one. In drug development, we are also trying to improve what we have in terms of infrastructures, from cell lines to micro-PET-CT for preclinical studies for small animals. Next. There are some, okay, so we are combining positron emission and gamma emission radioisotopes with therapeutical emissions, and in this regards we are of course looking forward to enlarge our range of radioisotope for therapies like Auger electron or alpha emitters. Next and next. There are some, yes, and next there are some animations here, but you know how the animal imagings are looking. I still have some slides.
Mm-hmm.
Okay, you can also put some double click on that.
Sorry, it's just jumping. The presentation is continuously jumping back to the first slide. I'm sorry about that.
Okay, I will speak in the meantime, because I just choose to show some examples of our work, and I of course choose copper-64, which is one of the true theranostic radioisotopes we are producing and investigating in our center. Next, please. And we also try to demonstrate that copper is also working as in its ionic form, and for that we are investigating a lot on radiobiology. Not only DNA targeting, where we know there are big damages caused by electron Auger, but we are also trying to investigate indirect effects, which it's presumed to be responsible for about 70% of the damages. And those we are, next please, we are investigating on different cell lines. And next, trying to assess cellular viability, DNA damage, and oxidative stress. Yes, next. For the production, we are using a fully automated station. Next. When we are including target material preparation of the solid target by electrodeposition, irradiation in our variable energy cyclotron, which allow us for a very nice and precise yield. Next one. Then we are doing dissolution and purification of our solution. Next. And in the final, we can get to a very good yield, as I say, up to 70 gigabecquerel of copper 64. Next, and the next, please. Just to exemplify what we are evaluating in terms of radiobiology, there are some slides, some graph showing the results in apoptosis and necrosis induced by radioactivity, and there are huge differences between them. We are also investigated other markers like oxidative stress markers and also LDH release induced by radioactivity. Next. Being just to make a link with the subject of the webinar, I was trying to put some questions about my during my professional paths. And next, please, there are a lot of, yes, you can go on, just go on with that. So one of that is that doing science in a developing country, and yes, that was a major challenge. Working in nuclear field, yes, we have to deal with regulation, restriction, and discipline, and commissioning of such a facility, what really involving a lot of burden. But what I wanted to stress is that for each of these challenges, we can get with some positive things, which are actually listed here in red. Very nice words like passion, dedication, and all of them are really true. Or everything is what I feel during my time. Go on with the next one. Because if you are looking at those words, they are all about science. It's about all that science needs, and there are also, in the meantime, women attributes. So I really think that supporting women to choose a career in science is not all about the gender balance, but it's more about equal opportunities or equal chances for all, and using the difference to make a difference, and using our strengths to make it better. So with this, I would like to thank you for your attention. Go to next to see our girl in the team, which are saying also hello to you.
I'm stuck again with the voilà.
Okay, that one. Okay, with my beautiful girls, yes. Thank you very much.
Thank you so much, Dana. Thank you for this inspiring talk. And then now we move to a younger, early scientist in her career, Kat Spoorman. Please, Kat, you have the floor.
So hi everybody, I'm Kat Spoormans. I'm a PhD student at SCKCN in Belgium, in collaboration with KU Leuven. I'm currently in my second year of my PhD research, which is about tumor control probability modeling in targeted radionuclide therapy, in which I will now give a short introduction. Next, please. So as you know, in radionuclide therapy, a ligand is coupled to a specific radioisotope. And this radiopharmaceutical is administered into the patient with a certain activity. Next, please. And it's the ligand which will determine first the activity distribution within the patient. So this activity distribution is a distribution of the isotopes itself, and this can be measured with, for example, a beta-word SPECT scan. Next, please. Then the selected isotope will determine the resulting dose distribution. The isotopes will emit particles with a certain energy, and they deposit their energy a bit further away from the isotope itself. And depending on the selected isotope, this range can differ, and so also will result in a different dose distribution. This dose distribution cannot be measured directly, but should be computed with computer simulations which simulate the interaction of the particles with the material in combination with relevant patient phantoms. Next, please. However, it's this dose distribution which we can't measure, which is in direct relation with the tumor response and the normal tissue toxicity in which we are interested. This relation between the dose and the responses is tried to be modeled with the so-called tumor control probability, or TCP model, for the tumor response on the one hand, and with the normal tissue complication probability, or NTCP model, for the normal tissue toxicity. In my research, I will focus on the first one, on the TCP. And such a model will need to include specific radiation characteristics, such as radiosensitivity, dose rate, dose heterogeneity, linear energy transfer, proliferation, etc. Next, please. So why is such a tumor control probability model important? It can be very valuable in evaluating and selecting, for example, in first stage different isotopes. When for a certain ligand the activity distribution is known, then we can, with such a model, evaluate the different isotopes, what the difference will be in tumor response and normal tissue toxicity, and this way have a first idea on the differences. And in a further stage, we hope it's also viable to go towards more individualized therapy, since now still fixed activities are given to the patients. Next, please. Now I will give you a short introduction on how such a TCP model itself looks like. Therefore, we need to start with probability for a single cell to survive the treatment, which is generally explained by the linear quadratic model. If you look at the cell survival in function of the dose, you see that at lower doses there's a linear behavior, but at higher doses a more quadratic behavior is observed, and this is generally interpreted as a superposition of two independent ionization events that superimpose to induce a lethal damage, which gives then a quadratic behavior. Next, please. The next step is to go to the probability that all the cells are treated in the tumor, which is then the TCP, the tumor control probability, which will have a behavior as you see here. Next, please. These models, as I show in those two slides, are already well established for external beam radiation therapy and used a lot during treatment planning. But in radionuclide therapy, as I told you, there's still no individual treatment planning. But radionuclide therapy would benefit a lot also from those models. But we cannot copy them as they are, but they should be adapted to the specific radiation characteristics of radionuclide therapy. And the main differences come in the dose rate, the dose heterogeneity, and the linear energy transfer. Next, please. So, for example, how we can incorporate the difference in dose rate. In radionuclide therapy, there's a much lower and protected dose rate, and this makes that in between two ionization events, the cells will have much more time to repair their DNA damage, such that there's less probability that two ionization events will combine to a single lethal damage, and which will be observed in a more linear response, as you see here. The quadratic part will vanish, and this then at the end results in a reduced efficacy also. Next, please. Another effect is a linear energy tensor, or LET for short. This quantity defines the density of the ionization track. You see that for alpha particles, they have a very dense ionization track, and it's also a high LET. Beta particles have a much lower LET. And these high LET particles will induce much more complicated DNA damages and are thus more effective, which is observed in a more steep survival curve. Next, please. So these were two examples of radiation characteristics that can be incorporated in such a DCP model. We are looking at more, and this is an overview of my research, how I will investigate these DCP models. I will look at it in an in vivo model, and this on the one hand requires very accurate dosimetry to really look at the dose response instead of only at the activity. And on the other hand, we will also look in vitro to input data for information about radiosensitivity and the repair mechanisms, etc. So this was my project in a nutshell, and I hope you enjoyed it.
Thank you. Thank you very much, Kat. Thank you. So now we have finished our 3 scientific presentations, and we have chosen for you 3 selected topics that are of importance and that we would like just to share some thoughts that we generated during the preparation of this webinar. So the first topic will be how to promote diversity, inclusion, mentorship and networking, and Renata will take the lead for this first topic. Then the second topic will be led by Ulla on ongoing efforts to bridge the gender gaps and to promote equal representation. And then I will take care of the third point, which is the potential impact of gender-related health issues when working with radiopharmaceuticals. So first topic, promoting diversity, inclusion, mentorship, and networking. Renata?
Yeah, thank you, Sarah. You all know that our field, radiopharmaceutical sciences, is very multidisciplinary field. So to work in that field, we need a lot of information and support from various disciplines, and this cannot be carried out without nice and good mentorship, networking, which in fact results in a efficient partnership. This has been recognized already by several institutions, and the need for proper support, mentorship, etc. is recognized by the women in radiopharmaceutical sciences, but also in women in nuclear, in associations like Women in Science. In the Society of Nuclear Medicine and Molecular Imaging, there is a group related to mentorship in nuclear medicine and radiopharmaceutical sciences. Similar activities are offered by EANM. I mentioned already today, Women's Empowerment group at the EANM. So there are many options, and you can always refer to older, more experienced researchers in the field for support in these areas. But do not forget the opportunities which are coming from attending the conferences and workshops, because in my understanding, this is the best way to directly communicate with your mentors. And we have so online communities, forums, grant and scholarships, and programs like COST, which is also supporting young researchers. So there is a lot of things and a lot of tools that can be used to promote diversity, inclusion, mentorship, networking, and partnership. Thank you.
Thank you, Renata. Second topic is ongoing efforts to bridge gender gaps and promote equal representation. And I would like to ask Ulla to take the floor.
Yeah, thank you. Yeah, in general, on bridging the gender gap, there is a lot going on a European level, particularly also on the highest level in the European Commission from our President Ursula von der Leyen, which made gender equality as a major theme and objective for the work of the Commission. And with this, we have issued a gender equality strategy for 2025, where, because when you address gender equality, you need data. You need data to show where we stand. And that's why, in order to address these gender gaps, also we at the JRC, I'm leading the directorate for nuclear, but we have colleagues working in other areas, and they are delivering a lot of data in the different fields, because you can only tackle gender issues if you have a good baseline where you can compare and benchmark. So this is important, the different areas in life to be tracked, so that you can also see how the trend is. And we also have seen that in many areas, the trends over many years was pretty stable, meaning not going up. Then that's why it triggered that more activities needed to be done. And one of the areas was, for example, women in boards of companies. This was long blocked because there were arguments that it is not needed because it will auto-regulate itself. After 10 years of discussion, we could show no, nothing had happened. The figures have not become better over 10 years. And the fact that we could demonstrate this led then to a regulation. So you need to foster these regulations. For Horizon Europe, there are gender equality plans, which need for the research team also to promote the gender balance and in the leadership positions. The equality bodies, where there is this European Institute for Gender Equality, which looks really to promote this in different areas. On the European Parliament, there is a Committee on Women's Rights and Gender Equality, and the European Association of Nuclear Medicine, which has established a gender diversity task force, and of course, which also includes then on nuclear medicine and radiopharmaceuticals. But I think it is probably even more pronounced in this field, because it was said before that it covers so many different areas, and bringing these different areas together makes it even more difficult to foster then the gender balance. But I think, on the other hand, this is also a strong point of this field, because it shows that you can reach out to so many other areas. And when you talk about gender equality, one point to reach this is also about visibility. Visibility of the colleagues working in this field in general, I would say, not only the female colleagues, but the importance of the field. Because then, when you have the importance of the field, you have also the purpose-driven people who want to join it. And also there are statistics that women have a lot of this purpose-driven background and want to go for it. So this could be another way, because it is also about raising the interest for the field and helping the young researchers to go into this area, because the competition for skills is everywhere. I mean, every field is now trying to attract good colleagues and students. So I think this is also something you have to start very early with gender equality, by attracting the right students. And we in JRC are also trying to help with this by giving visibility to this by organizing high-level workshops on the different aspects for radioisotopes. And also we will have in the autumn several events coming up, and I will also post on these different events, and I would also invite colleagues to join, because this kind of events where you can discuss with others, as it was said before, by the networking, you have to link with others, you have to be in contact, and that helps you then to get to other positions. Thank you.
Thank you so much, Ulla. Very inspiring presentation as well. And then the final topic that we have for today is the potential impact of gender-related health issues. A few points that we would like to also bring on this platform is of course, the importance of medical surveillance. There are in Europe, in research institutes, university clinics, robust medical surveillance programs that considers gender-related factors. And in terms of personal protective equipment that are in some cases specially designed, such as lead aprons and shielding to accommodate different body shapes and different sizes. So there we see also an evolution to protect all genders and with all figures and without compromising the comfort and the safety. We would like also to mention a topic which is very close to certainly the early career scientists. It's the reproductive health. We know that female workers of child-bearing age are usually kind of isolated from the lab during the period of the pregnancy. And this is, of course, for a good reason to have like special attention and special protection due to potential risks during the reproductive health, since during the embryonic development there is a higher risks for the embryo for adverse effects following exposure. So there are strict protocols that are and guidelines that are implemented for pregnant or potentially pregnant employees to minimize fetal exposure. Maybe also some other topics that we thought would be good to bring forward was the difference in individual radiation sensitivity. Different genders might exhibit varying levels of sensitivity to radiation exposure. We see that in radiotherapy treatment between patients, so studies clearly suggest that females could be more sensitive to ionizing radiation, and mainly due to physiological and genetic differences. So it is really important to continue to ensure proper monitoring and safety measures to mitigate potential gender-based variations in radiation response. Training, education are really important to provide comprehensive training that also covers gender-specific health considerations, to also ensure that all the workers are well informed about potential risks and safety measures, with and then reporting and support, so to really encourage open communication about any gender-related health concern. Being able to discuss their unique health needs, also ensuring support and adjustments where needed. Another topic we would like to mention is data collection, really to continue to promote research and into potential gender-based differences in radiation response and in health outcomes. This is also something which is put on the agenda at the European level, but it needs to continue to be developed. So to collect and to analyze data to refine safety protocols and to address any disparities. And inclusivity and diversity, so to really continue to foster an inclusive workplace culture that values diversity and that recognizes also the potential impact of gender-related health issues. So we thought that addressing all these points can certainly contribute to a safer and a more equitable work environment when working with radiopharmaceuticals and considering the potential impacts of gender-related health issues. So this was a bit the message that we wanted to give you today during this webinar, and of course we are now open and here also to listen to your questions, any comments, any issues that you would like to raise. So you have the chat box. Please use it and add your comments and any remarks that you would like to express. So I've seen that on the chat, that in the chat box we have all the information for the ones who would like to join the PRISMAP call for proposals. Do not hesitate to really reach out, ask questions on the help desk, and from the moment you have an ID, you receive an appointed mentor that will help you in the process and in the process of preparing your presentation. So do not hesitate, and the PRISMAP project is really there for you. Any remarks from the panelists? I don't know, Renata, Ula?
Yeah, just wanted to add, because Thierry was sending a short message on the chat about production of radium-224, but in this short message he didn't mention that it's his female researcher colleagues working on this production. So we congratulate them, and we understand that they cannot follow the webinar because they are busy in the lab, which is very often the case for many of us. And I would like to express my gratitude to our speakers today for their presentations. Very nice and very interesting presentations. And we would like to learn more about your research in the future. Thank you.
Thank you. Thank you so much. Thank you everyone. Thank you all the panelists for this very inspiring and interesting, fruitful presentations. And we hope that this webinar was useful for you, and do not hesitate to contact us. And thank you, Renata, for having co-moderate this webinar together with me. So if no question, bye-bye to everyone. Have a lovely weekend.
Thank you.